Cardiac-derived cells are placed onto cell culture plates for a simulated ischemia reperfusion model. Ischemia is achieved with 150 minutes in a glucose-free medium while the oxygen level is below 0.5%. This ischemic period leads to the shrinking and blebbing of most cells. Then, normal media and oxygen levels are reintroduced to simulate reperfusion. Mesenchymal stem cells are added and cultivated together with the damaged cells. After 24 hours of co-cultivation, cells are stained with calcine and ethidium homodimer to differentiate between live and dead cells. Confocal microscopy clearly shows the two cell populations and their interactions. Flow cytometry analysis reveals 3 clusters of damaged cells which can be plotted on a graph and analyzed statistically.
Stem cell transplantation protocols are finding their way into clinical practice, getting better results, making the protocols more robust, and finding new sources for implantable cells are the focus of recent research. Investigating the effectiveness of cell therapies is not an easy task, and new tools are needed to investigate mechanisms involved in the treatment process. We designed an experimental protocol in order to allow the observation of cellular connections and even subcellular mechanisms during ischemia reperfusion injury and after stem cell transplantation. Our design criteria were fully in vitro system where only the host and the donor cells are present, so it is isolated from the surrounding tissue. Continuous measurement of cell viability before, during and after the injury for up to 24 hours. And clear identification of both cell populations so the fate of the grafted cells can be traced. In order to reach this goal, we set up a protocol of oxygen glucose deprivation, which is commonly abbreviated as OGD, followed by a reoxygenation period. Our choice of host cells was H9C2 cells, a well-known red cardiomyoblast cell line. After OGD,